All right, we're going to talk about photosynthesis and how the sun is the ultimate power source for all living organisms on the entire Earth. Some terms you need to be familiar with, and I'm sure you're probably already familiar with, the first one is photosynthesis, and that is how we convert light energy into chemical energy. Autotrophs are organisms that are able to make their own food, and if we refer to a photoautotroph, we're speaking about an organism that uses sunlight to make their own food. Heterotrophs obtain their organic material from other organisms, so they eat autotrophs or other heterotrophs, and just about all heterotrophs, including us humans, depend on photoautotrophs or plants for food and oxygen. Common misnomer about photosynthesis is that it only occurs in plants. Photosynthesis actually occurs in plants, algae, some protists, and some types of prokaryotes such as cyanobacteria, which you see on the bottom. Chloroplasts are what make photosynthesis possible. Remember that these are structurally similar to photosynthetic bacteria. Most likely, they actually were at one point an independent living bacteria that was photosynthetic, and we remember that from the endosymbiotic theory. The green color of photoautotrophs comes from chlorophyll, which is a pigment molecule found in chloroplasts. Light energy is absorbed by the chlorophyll, and that's what drives the synthesis of organic molecules such as glucose, and that happens in the chloroplast. CO2 enters the plant, specific to the plant, through the stomata, and oxygen leaves through the same stomata, and water also leaves, which is a product of photosynthesis. The mesophyll is the interior layer or tissue part of the leaf, and that's where you're typically going to find your chloroplasts. The chlorophyll inside the chloroplast is found in a thylakoid, and this looks like a flattened disc. And if you get a bunch of thylakoids and you stack them on top of one another, it looks like a stack of pancakes, or a thylakoid stack that we call a grana. Inside the chloroplast, there is a dense fluid similar to the cytoplasm or the mitochondrial matrix, and that is called the stroma. And here's some pretty pictures to go along with that. This interior layer right here is the mesophyll. That's where you find your pigment molecules. Here are your thylakoids. One single thylakoid looks like a little Mento on top. And if you get a stack of thylakoids, this is considered a grana. And those are found in the chloroplast, which has your chlorophyll in it. All right, the equation for photosynthesis is very similar to the equation for cellular respiration, just reversed. So we start out with 6 carbon dioxides, 6 waters, plus light energy, and we form a glucose molecule or some sort of organic molecule, as well as oxygen. The electrons that we get come from the splitting of water, which is an endergonic reaction. This is powered by the sun. So the sun provides the energy for this reaction that has a positive Gibbs free energy to occur. Chloroplasts split water into hydrogen and oxygen atoms, and the electrons from that water molecule are then incorporated into glucose or sugar molecules. So from this, we can see our reactants and where they go. Carbon dioxide becomes part of the glucose molecule. Part of the oxygen also forms water, and we didn't see this in our last reaction, but water is also a product of photosynthesis. We use 12 molecules of water and we produce 6. So we simplify the equations to show that there isn't any water made and that we need 6 water molecules in order for photosynthesis to occur. So there is some water that is produced. It was not shown in the last equation. Water split forming oxygen molecules. That water is used also to form some of the water that is product, and it contributes some hydrogen ions to that glucose. So remember from cellular respiration, we talked about oxidation and reduction reactions. Photosynthesis is also a redox process. Water is oxidized, it loses electrons, and CO2 is reduced. It gains electrons to help form glucose. Photosynthesis has 2 main steps. It has the light reactions, which is the photo part, and the dark reactions, or the light-independent reactions, which are called the Calvin cycle. And this is the synthesis part, where we actually make our glucose and our organic molecules. Light reactions convert the light to chemical energy. This happens in the thylakoids. The reactants are water, and when water splits into hydrogen and oxygen, that's where you get your electrons from. You need to start with an electron carrier, NADP+, and you also need a DP and an inorganic phosphate. We produce, through results of the light reactions, oxygen, NADPH. Okay, so that is the electron carrier that has picked up 2 electrons and picked up 1 hydrogen ion, and ATP, which happens from photophosphorylation. So that is using sunlight to take your inorganic phosphate and add it to ADP. As we go through these, keep in mind these 3 main points for each step. Where does it happen? What does it need to start? And what is produced? Because photosynthesis is a cycle, and the reactants or the products of one cycle are usually the reactants for the next. The Calvin cycle, also called the dark reaction, also called the independent light reaction, begins with carbon fixation. So this is where we take our carbon dioxide molecule and we create a glucose molecule, or a C6H12O6 molecule. This happens in the stroma. We need CO2, ATP, and NADPH to begin. That ATP and NADPH came from our light reactions. So those were the products from the light reactions that become the reactants for our dark reactions. We produce sugar, which will leave the chloroplast to where it's needed in cellular respiration elsewhere in the cell. That ATP will be phosphorylated, so it splits into ADP and inorganic phosphate. Those will get reused in the next, as it cycles back to the light reactions to form ATP. And our electrons get dropped off, so your electron carrier is now empty, which will cycle back to the light reactions. That was a lot of talking, and now here's a pretty picture. So we need water to start the light reaction. That's the first stage, and it is light energy that splits water and allows us to get those electrons, which are picked up by your electron carrier. NADP plus picks up your 2 electrons to form NADPH. This travels to the Calvin cycle. That is a reactant for the Calvin cycle. And we also use ADP and inorganic phosphate to produce ATP, which is a reactant needed for the Calvin cycle. The Calvin cycle uses CO2 to form sugar, and it also takes those 2 electrons from the electron carrier and uses the energy stored in ATP, and it spits back out the sugars, which are used elsewhere, and then ADP and inorganic phosphate and your empty electron carrier, which are going to be used to start the light reactions again. So these guys, think of them as a cycle between the 2 reactions. The reactants of one are the products of the other. So it's your thylakoids inside the chloroplast that are transforming light energy into chemical energy in the form of ATP, and also carrying our electrons, your NADPH, your electron carrier. Light is an electromagnetic energy. The light energy does travel in waves, but it acts like it's made out of discrete particles called photons, kind of funky like that. Wavelength, remember, is the distance between crests of waves. So if you have a wave like this could be light. Wavelength is the distance from one crest to another, or one trough to another. And your wavelength is going to determine the type of electromagnetic energy. Closer, or the shorter the wavelength, the higher the energy. The longer the wavelength, the less energy that it carries. So it is important to remember that visible light is part of the electromagnetic spectrum, which includes gamma rays, UV light, microwaves, radio waves. It is a portion of that, and it is right in the center. Your visible light is wedged between your UV and your infrared. And the closer you get to UV light, or purple, the shorter your wavelength and the higher the energy. And the further away from UV towards the red end of the visible light spectrum and your infrared side, the longer the wavelength and the lower the energy. In photosynthesis and the light reactions, it's all about the pigments. And these are the molecules that are going to absorb light. So if you have different pigments, those absorb different wavelengths. If the wavelength is not absorbed by that pigment molecule, that wavelength is then reflected, and that is the color that we see in the visible light spectrum. So in chlorophyll, the leaves and chlorophyll itself appears green because it's absorbing all wavelengths of visible light except for green. Green is being reflected and transmitted. Just in case you didn't believe me about that, here is a picture. Visible light is made up of red, orange, green, blue, yellow, violet, and all of those colors are being absorbed by the thylakoids, by the pigment molecules in the thylakoids in the chlorophyll, and green is not. So green is reflected back, and that's why plants look green. One of the ways that we can measure wavelengths and what's being transmitted versus absorbed is by using something called a spectrophotometer. And a spectrophotometer measures a pigment's ability to absorb various wavelengths. So the way that we set this up is we get a prism that's going to separate your visible light into the spectrum, and we put a filter on there so that it's only green light is passing through. And then we take a chlorophyll solution, pass the green light through the chlorophyll solution, and we look at what the light transmittance is. And this particular setup, because of the green light, chlorophyll does not absorb green light, so it has a very high transmittance of light, meaning the green light travels straight through the chlorophyll. It's reflected, very low absorption. In this particular setup, we move the slit, the opening, so that only blue light is passing through our chlorophyll solution. We know that chlorophyll absorbs blue light, so we have very low transmittance of light through the chlorophyll solution, or a very high absorption. So that tells us that chlorophyll is absorbing the wavelength that is blue light. So if we were to take our spectrophotometer data and put it into a beautiful, colorful graphical form, this is what we'd get, which is Engelmann's experiment. So we see that the highest points of absorption for wavelength happen in the violet blue range, as well as in the red range. We get our highest absorption. So those tend to be the wavelengths of light that are most important for photosynthesis. If we were to look at a plot of absorption versus wavelength, we can look at the rate of photosynthesis. So here we see highest photosynthesis happens right here in the same region that we saw our peaks in. So this just shows us that photosynthesis tends to occur when you are exposing your plants or your chlorophyll to that red wavelength of light, as well as that violet blue wavelength of light. Chlorophyll A is your main photosynthetic pigment. You have also accessory pigments such as chlorophyll B and carotenoids that assist in absorbing other wavelengths of light. So chlorophyll B broadens that spectrum, so it increases the range of wavelength that can be used for photosynthesis. Carotenoids are what you find down in that red-orange area, that second big peak that we saw, and these guys absorb excessive light that would otherwise damage the chlorophyll. They're also what make leaves in the winter, as the fall starts to come around, that's what makes the leaves look orange. So your pigment molecules are embedded in the thylakoid membrane, which means they're going to need to have a head that can be exposed to water, and a hydrophobic tail, because this part is actually embedded in the membrane, so it also has to be hydrophobic. Your chlorophyll A and chlorophyll B are structurally on there, and we have a center head made of magnesium. So we use light to power the excitation of electrons. So light has a lot of energy. It hits your pigment molecule, your chlorophyll, which excites the electrons, so they go from a ground state to an excited state of electrons. And if those are not captured and put to work within photosynthesis within your chlorophyll, those electrons are going to drop back down to their ground state. And as they fall back down, they're going to give off energy in the form of heat and light. And so what you're looking at right now is the fluorescence of chlorophyll molecules, of electrons in the chlorophyll molecules that have been excited. So they're fluorescing right now, giving off both light and heat. Photosystems consist of both a reaction center complex as well as a light harvesting complex. In the light harvesting complex, you're going to have a photon that hits your light harvesting complex, and it's going to excite electrons. And those electrons are going to bounce around in that until they hit the reaction center. Once they hit the reaction center, that's when they pass those excited electrons to the primary electron acceptor. It is at this point that your light energy from the photon that turned into those high energy electrons, excited those electrons, gets converted into chemical energy. So here's the pretty picture. Your photon of light is going to hit a pigment molecule in the light harvesting complex. That electron is going to bounce around in the light harvesting complex until it comes to the reaction center. This portion, this lighter purple, is the reaction center. Once the electron hits the reaction center, it is then passed to the primary electron acceptor, the P. Once it's in the P, it has been converted completely from light energy to chemical energy, and we can do something with that energy. We can harness it to do work now. Thylakoid membranes have not one, but two photosystems that act in cooperation with one another. Photosystem 2 functions first in photosynthesis. It's named 2 because it was discovered second, although it functions first. It is best absorbing a wavelength of 680 nanometers, so therefore we call it P680. Photosystem 1 functions second and is best at absorbing a wavelength of 700 nanometers, so we call it P700. When an electron gets excited during the light reactions, there are two routes that electron can flow through. It can either go through cyclic electron flow or linear electron flow. Linear or non-cyclic electron flow is your primary pathway, and it involves both photosystems. So moving from photosystem 2 to photosystem 1, it produces ATP, and your electrons are picked up by the electron carrier NADP+ to form NADPH. This uses light energy. Okay, so visually here's how it works. You have a photon of light that's going to hit the pigment molecule. Your electron bounces around excited until it hits a P680 pigment molecule. The excited electron is passed from P680 up to a primary electron acceptor. At this point, your P680 is now missing an electron. Okay, it's a very strong oxidizing agent. So water is split into hydrogen and oxygen ions, and in that split, it replaces electrons to the P680. Okay, and because it accepts electrons in the P680, it has been reduced. Oxygen is produced as a byproduct of this. So that electron that gets passed to the primary electron acceptor has to be replaced by another electron, and that electron comes from water. So moving from photosystem 2 to photosystem 1 requires a bridge, and that bridge is an electron transport chain. The primary electron acceptor moves an electron into the electron transport chain. As it moves from protein to protein through the electron transport chain, energy is released each time. This energy is used to drive the movement of hydrogen ions or protons across the thylakoid membrane. It creates a gradient, so it's moving hydrogen ions from low concentration to high, so it's making a very high concentration on one side. Those guys are going to want to move back through the membrane. When they diffuse back through the membrane, that is what drives ATP synthesis, just like in cellular respiration. Here's where photosystem 1 comes in. This electron eventually is going to wind up in the P700 area. Remember that light hits the P700, or light with the wavelength of 700 nanometers, is going to hit that pigment molecule, and it's going to excite an electron that's going to bounce around until it hits P700. From P700, it goes up to a primary acceptor of electrons. Once it has moved to the primary electron acceptor, it needs an electron to take its place, and that electron comes from the electron transport chain, which brought the electron over from photosystem 2, which the electron had originally come from the splitting of water. So we go from water to PS680, primary electron acceptor, through the electron transport chain to replace the lost electron at P700. Eventually, our big goal of moving those electrons around and getting them excited is to get those picked up by an NADP+ molecule, an electron carrier. Once they are picked up by NADP+, we also pick up a proton or hydrogen ions. We pick up two electrons, hydrogen ion, to form NADPH. Because it has accepted and gained an electron, we consider it to be reduced. We are going to take these electrons that were excited, these high energy electrons excited by the light reaction, and we're going to transfer them over to the Calvin cycle where we can put them to work. So our last step, we have that electron that's moved through the photosystems. It is taken up by the primary electron acceptor, and what do you know? It enters another electron transport chain. At the end of this electron transport chain is an enzyme that allows NADP+ to pick up a hydrogen ion as well as two electrons. So here's an analogy to show us why we have two photosystems. In your first photosystem, a photon charges up your electron so it has an excited state. So it's at about this level. As it moves from one photosystem to the other, it is going to lose energy because it is powering the synthesis of ATP. It's powering that proton gradient that drives ATP synthase to form ATP. When it gets to the second photosystem, it has lost some energy. And if you look right here, we can see that it's lost approximately that much energy from when it was first charged to after it's made some ATP. What we do is we then recharge it with another photon of light. So we get it up to a level, an excited level that is much higher than that original level. As you can see, photosystem 1 only gave it so much energy, and now we can recharge it to get even a higher level of energy than what we had, and we've made some ATP in the process. At this point, that electron, that excited electron, is ready to be accepted by NADP plus and picked up by that electron carrier, and then it can be taken over to the Calvin cycle where those electrons can be harnessed to do work in creating sugars. So we talked about linear electron flow, where you are moving from one photosystem to the next and you're creating some ATP. In plants, there's also cyclic electron flow. And in this case, cyclic electron flow, once your P700 picks up that electron, it goes to the primary electron acceptor, it can actually travel back to the electron transport chain to continue providing energy to regenerate more and more ATP as needed through the Calvin cycle. So not all of the electrons are going to leave the primary electron acceptor to join up with your electron carrier NADP+. Organisms such as purple sulfur bacteria have a photosystem 1, so the second part, but not the photosystem 2. And what this led scientists to believe is come up with this idea that cyclic electron flow evolved before linear electron flow. And they think that cyclic electron flow protects cells from light-induced damage. So you continue reusing those electrons and recharging them from one wavelength. Having only a photosystem 1 and not a photosystem 2 means that the only way you're going to generate ATP for photosynthesis is through cyclic electron flow. So it does produce ATP by chemiosmosis, so creating a proton gradient and having those protons flow back through ATP synthase, but you don't produce any NADPH, and therefore no oxygen is produced. A lot of what we're talking about in photosynthesis is kind of a repeat of what we've seen in cellular respiration. Chloroplasts and mitochondria are both responsible for generating ATP by chemiosmosis, but they use different sources of energy to do this. Mitochondria are transferring your energy from food or glucose to ATP, whereas your chloroplasts will transform light energy into chemical energy to form ATP. In the mitochondria, you have protons that are pumped to the inner membrane space, and as they diffuse back across the membrane, they flow through ATP synthase, and they produce ATP in the mitochondrial matrix. In chloroplasts, same idea, different location. Your protons are pumped into the thylakoid space, and they drive ATP synthesis as they diffuse back into the stroma of the chloroplast. ATP and NADPH are produced on the side facing the stroma, where the Calvin cycle takes place, because they are used in the Calvin cycle, so they need to be in close location to the Calvin cycle. Location's different, idea is the same. So whether we're talking about the inner mitochondrial membrane or the thylakoid membrane, you are still pumping hydrogen ions across from a low concentration to a high concentration. You're using an electron transport chain to provide energy to get those pumps to actively transport hydrogen ions. They want to diffuse back across the membrane. They want to reach equilibrium again. The only place they can go through that membrane to reach equilibrium is through ATP synthase, which is an enzyme that produces ATP from ADP and inorganic phosphate. So the idea, the mechanism is the same, the location in the cell is different. If we were to sum it up in one sentence, your light reactions generate ATP. They increase the potential energy of electrons as they move them from water to the electron carrier NADPH. All right, so if we were to look at the light reactions in a nutshell, this is what they'd look like. You have photosystem 2, where light first excites electrons. It bounces around till it hits P680 and is accepted into the primary electron acceptor. It travels through the electron transport chain. While it's traveling through there, it is powering hydrogen ions getting pumped into the thylakoid space. So we're increasing the hydrogen ion concentration because eventually They're going to flow out of the ATP synthase, forming ATP, which will be used in the next step, which is the Calvin cycle. The electron has a little less energy. It enters photosystem 1, where it sits in photosystem 1 until light energy further excites it. It enters P700, that's the pigment molecule, enters a primary electron acceptor. It is passed to another electron transport chain, which drops it off at the electron carrier NADP+. 2 electrons and 1 hydrogen ion are picked up, forming NADPH, which will be used in the Calvin cycle. So the Calvin cycle is similar to the citric acid cycle in cellular respiration because it regenerates its starting material after molecules enter and leave the cycle. The cycle will build sugars from smaller molecules, and it uses ATP and the reducing power of electrons carried by NADPH in order to accomplish this. All right, carbon will enter the Calvin cycle as carbon dioxide, and it leaves as a sugar called glyceraldehyde 3-phosphate, or G3P for short, which we can then use to make glucose and other organic compounds. If we want to make one G3P, the cycle has to take place 3 times. So it has to fix 3 molecules of CO2 in order to make one G3P. It has 3 phases. There's carbon fixation, reduction, and the regeneration of the CO2 acceptor, RUBP, which is needed to begin that again. Okay, this is daunting. What you really need to know is what is coming in and what is leaving. So here we have 3 CO2s entering. They react with Rubisco to form intermediates. We are going to use energy in the form of ATP in order to add carbons onto this chain. You are going to drop off those high energy electrons. Now you have an empty electron carrier, which is going to travel back to the light reactions to pick up more electrons. You will form G3P, which is a sugar. This can be used to make glucose and other compounds. This is the reduction phase, when you are picking up those electrons. And I didn't mention this is carbon fixation. You're taking carbon from the atmosphere and making these longer carbon molecules. Reduction, you pick up your electrons. This G3P is going to move out. It is a sugar. It can be used in cell respiration. However, we have to reform Rubisco. We have to reform ribulose bisphosphate, RUBP, so that we can make this cycle go again. So the last part, the last stage, is the regeneration of your CO2 acceptor, RUBP. And this also requires some energy. You have to have this back again to start your cycle over. In summary, we need light and water to start our light reactions. Your electrons from water are going to travel through photosystem 2, the electron transport chain, and photosystem 1. In that time, you are going to produce some ATP, which are going to be used in your Calvin cycle, and you're also going to pick up high energy electrons, which will be used in your Calvin cycle as well. A byproduct of this is oxygen. Using CO2, which is another product, CO2 reacts with Rubisco and picks up high energy electrons to form sucrose, or G3P, which can eventually be turned to sucrose. And then in the last stage, it regenerates that starting material that accepts CO2. So this is how we get sugar.